Hello and welcome to your ultimate guide for the D312 OA, Anatomy and Physiology course. My goal today is simple: give you everything you need to fully understand the key concepts, master them, and walk into your D312 OA exam with total confidence. In this video we'll cover nervous systems, brain anatomy, endocrine signaling, pituitary axis, special senses, and remember, our website offers free exam guides and practice questions to help you ace your D312 OA exam. You'll find the link in the description below, so be sure to check it out and get the resources you need to study smarter, not harder. So let's get started. The nervous system does three things. Sensation means it detects stimuli, whether that is heat on your skin or light hitting your retina. Integration means it processes and interprets that information in the brain or spinal cord. Response means it activates muscles or glands to do something about it. Every question about the nervous system traces back to one of those three functions. Your nervous system splits into two main divisions. The central nervous system, or CNS, is your brain and spinal cord. The peripheral nervous system, or PNS, is everything else, all the nerves running through your body carrying information to and from the CNS. Think of the CNS as headquarters and the PNS as the field agents. The PNS breaks down further into three parts. The somatic nervous system handles voluntary stuff like moving your arm. The autonomic nervous system controls involuntary functions like heart rate and digestion. And the enteric nervous system runs your gastrointestinal tract mostly on its own using chemoreceptors and stretch receptors to manage peristalsis. The autonomic nervous system has two branches you need to know cold. The sympathetic division is fight or flight. It speeds up your heart, dilates your pupils, and diverts blood to your muscles. The parasympathetic division is rest and digest. It slows your heart, constricts pupils, and promotes digestion. These two systems oppose each other to keep your body balanced. Before we get deeper into the CNS, I want to mention something that will help you study. I've created over 300 free practice questions specifically for D312 that cover all of this material. The link is in the description below. Bookmark it now so you can test yourself after watching this video. It makes a real difference. The brain has four main regions. The brainstem handles basic life functions. The cerebellum coordinates movement and balance. The diencephalon includes the thalamus and hypothalamus. And the cerebrum is where your higher thinking, memory, and voluntary actions happen. One fact that comes up on exams is that glucose is the brain's only energy source. Inside the CNS, tissue is either white matter or grey matter. White matter is made of myelinated axons, and the myelin sheath gives it that pale colour. Grey matter is where the cell bodies, dendrites, and unmyelinated axons cluster. In the spinal cord, grey matter sits in the centre, in a butterfly or H-shaped pattern surrounding the central canal. On the exam, if they describe a cross-section of the spinal cord and ask about that central region, grey matter is your answer. The grey matter houses interneurons and motor neuron cell bodies, while the surrounding white matter carries signals up and down the cord. Knowing which is where in the spinal cord is a straightforward point you do not want to miss. Three protective membranes called meninges surround the brain and spinal cord. The outermost is the dura mater, which is tough and thick. The middle layer is the arachnoid mater, a thin, transparent membrane forming a loose sac. The innermost is the pia mater, a delicate layer that lets blood vessels pass through to nourish brain tissue. Cerebrospinal fluid fills the space around the brain and spinal cord. It acts as a shock absorber and contributes to the blood-brain barrier. The choroid plexus produces it. If your exam asks what produces cerebrospinal fluid, that is the answer every time. Choroid plexus. Write it down, say it out loud, lock it in. The spinal cord exits the skull through the foramen magnum and runs through the vertebral column. It has 31 pairs of spinal nerves. Sensory fibres enter through the posterior dorsal roots. Motor fibres exit through the anterior ventral roots. A memory trick for this is that sensory is dorsal and motor is ventral. Dorsal is the back, ventral is the front. At the bottom of the spinal cord, things get interesting. The conus medullaris is where the cord tapers off. roughly at the first lumbar vertebra. Below that, the remaining nerves fan out into what is called the cauda equina, which means horse's tail in Latin, because that is exactly what it looks like. The filum terminale anchors it to the coccyx. Now for the cells themselves. Neurons have three parts: a cell body called the soma, dendrites that receive signals, and an axon that transmits signals away. The synapse is the gap between two neurons where chemical messengers cross over. This basic architecture shows up in almost every nervous system question you will face. The synapse itself is worth pausing on. When an electrical signal reaches the end of an axon, it triggers the release of neurotransmitters into the synaptic cleft. These chemical messengers drift across the gap and bind to receptors on the next neuron. That binding either excites or inhibits the receiving cell. The whole process takes milliseconds. Your exam may describe this sequence and ask which step is disrupted. Neurons are classified two ways. By shape, you have unipolar neurons with one process, bipolar neurons with two, and multipolar neurons with many. Multipolar are the most common type. By function, sensory neurons carry signals toward the CNS, motor neurons carry signals away from it, and interneurons sit inside the CNS, bridging the two. Those practice questions I mentioned earlier cover exactly this kind of classification material. The exam loves asking you to match neuron types with their functions or identify which direction a signal travels. Working through those problems is the fastest way to lock this down before test day. The link is right in the description. Neurons get all the credit, but glial cells do the real support work. In the CNS, astrocytes provide structural support. Oligodendrocytes handle myelination, which is insulation of axons. Microglia act as the immune cells, cleaning up debris. Ependymal cells produce cerebrospinal fluid. In the PNS, Schwann cells myelinate axons, and satellite cells provide support. The key exam distinction here is which glial cells belong to the CNS and which belong to the PNS. Oligodendrocytes myelinate in the CNS. Schwann cells myelinate in the PNS. Both do the same job of insulation, just in different territories. If you remember nothing else from this section, remember that split. It shows up repeatedly on the OA. Nerve structure in the PNS mirrors muscle structure in a way that helps you remember both. Individual axons are wrapped in endoneurium. Bundles of axons form fascicles wrapped in perineurium. The whole nerve is covered in epineurium. Endo is innermost, peri is middle, EPI is outermost. Same naming pattern as muscle tissue layers. There are 12 pairs of cranial nerves, and you need to know the big ones. CN1 is the olfactory nerve for smell. CN2 is the optic nerve for vision. CN5 is the trigeminal, which handles facial sensation and controls the muscles of chewing. CN7 is the facial nerve, controlling facial expressions and carrying taste signals. CN8 is the vestibulocochlear nerve, responsible for both hearing and balance. CN10 is the vagus nerve, which is the big autonomic nerve handling sensory, motor, and visceral functions across the chest and abdomen. CN6, the abducens, moves the eye laterally through the lateral rectus muscle. That connection between nerve and muscle comes up on scenario questions. I know we are covering a lot of detail here, and you might be wondering how to practice applying all of it. That is exactly why those free practice questions exist. They combine multiple concepts the same way your OA exam will. Cranial nerve questions, neuron classification, glial cells, all of it. Now we are shifting into the endocrine system, which works hand in hand with the nervous system. The endocrine system uses hormones, chemical messengers released by glands into the bloodstream. These hormones travel to target cells that have specific receptors for them. No receptor, no response. That selectivity is what makes the system precise. Two types of glands matter here. Exocrine glands secrete through ducts onto body surfaces, like sweat glands. Endocrine glands are ductless and release hormones directly into blood. When your exam says ductless gland, it means endocrine. When it says secretes through a duct, it means exocrine. That is one of those binary distinctions they love testing. Hormones come in two flavours based on solubility. Water-soluble hormones cannot cross the cell membrane, so they bind receptors on the cell surface and use second messenger systems inside the cell. Lipid-soluble hormones, usually built from cholesterol, pass right through the membrane and bind receptors inside the cell, often directly changing gene expression in the nucleus. The difference in signaling mechanism between these two hormone types is a frequent exam topic. Water-soluble stays outside the membrane and uses a second messenger relay. Lipid-soluble passes through the membrane and acts directly on the nucleus. Picture it like a locked door. Water-soluble hormones knock and send a messenger inside. Lipid-soluble hormones have the key and walk straight in. 3 things can trigger hormone release. Neural stimuli happen when nerve fibers directly stimulate a gland, like the sympathetic nervous system activating the adrenal medulla. Humoral stimuli happen when blood chemistry changes, like rising glucose triggering insulin. Hormonal stimuli happen when one hormone triggers another gland, like TSH from the pituitary, telling the thyroid to release its hormones. Hormones can also interact with each other in ways the exam tests. A permissive hormone is one that amplifies the effect of another hormone. An antagonist hormone blocks another hormone's receptor, preventing the expected response. Think of it like radio signals. A permissive hormone turns up the volume. An antagonist jams the frequency. Knowing these interaction types helps you answer questions about why one hormone seems to cancel out another. The pituitary gland is the centerpiece of the endocrine system, and it sits just below the thalamus. It has 2 lobes with completely different jobs. The anterior pituitary, called the adenohypophysis, is a true endocrine organ, making its own hormones. The posterior pituitary, called the neurohypophysis, stores and releases hormones that were actually made in the hypothalamus. The hypothalamic pituitary axis is the feedback loop connecting these structures. The hypothalamus monitors blood hormone levels and adjusts its releasing or inhibiting hormones accordingly. Those signals hit the anterior pituitary, which adjusts its own hormone output, affecting downstream target glands. When the target gland produces enough hormone, the hypothalamus detects the rise and dials back. This negative feedback loop shows up constantly on the OA. If some of these terms feel heavy right now, that is completely normal. The pituitary alone has enough detail to fill an entire chapter, and most students need a few passes to sort it out. Working through targeted practice problems helps you figure out which lobe does what and which cell makes which hormone. Those free questions in the description are built for exactly this kind of sorting. The anterior pituitary has 5 cell types, and each one produces a specific hormone. Thyrotrophs make TSH, which targets the thyroid. Corticotrophs make ACH, targeting the adrenal cortex. Somatotrophs make growth hormone. Lactotrophs make prolactin for the mammary glands. Gonadotrophs make both FSH and LH, targeting the ovaries and testes. The hypothalamus controls these anterior pituitary cells through releasing and inhibiting hormones. TRH stimulates TSH, CRH stimulates ACH, GRH stimulates growth hormone while GHIH inhibits it. GnRH stimulates FSH and LH. These hormones travel through a special blood connection called the hypophyseal portal system. That portal system name is a common exam question. The posterior pituitary works differently. It does not make hormones at all. 2 nuclei in the hypothalamus do the manufacturing. The supraoptic nucleus makes ADH, also called vasopressin, which tells your kidneys to retain water. The paraventricular nucleus makes both oxytocin and ADH. Oxytocin triggers uterine contractions and milk ejection. Pituocytes in the posterior pituitary are specialized glial cells that support the axon terminals storing these hormones. Blood supply comes through the posterior hypophyseal plexus. The exam sometimes asks what distinguishes the posterior pituitary from the anterior, and the answer is that the posterior pituitary has nervous function, not endocrine manufacturing function. Mastering these hormone pathways takes repetition, and that is where practice makes the difference. The free practice questions I put together walk you through each axis from hypothalamus to pituitary to target gland. They are structured to build that chain of reasoning your exam expects. You will find the link right in the description below. Now we are moving into the special senses, starting with vision. The eye wall has 3 layers called tunicae. The outer fibrous tunica includes the sclera, the white of the eye, and the cornea, the clear dome that refracts light. The sclera is avascular and maintains the eye shape. The cornea is also avascular and is where light first bends. The middle vascular tunica has 3 parts. The choroid is a brown layer packed with blood and lymphatic vessels. The ciliary body sits behind the iris, secretes aqueous humor, and adjusts lens shape. The iris is the coloured part with 2 rings of smooth muscle that control pupil diameter. The constrictor muscle to shrink the pupil and the dilator muscle to widen it. The innermost neural tunica is the retina. It contains 10 layers of cells, including the photoreceptors that actually detect light. The macula lutea is where your clearest vision happens, packed with cone cells. At its centre, the fovea centralis gives you your sharpest focus. The optic disc is where the optic nerve exits, and it has 0 photoreceptors, making it your blind spot. 2 types of photoreceptors do different jobs. Rod cells work in low light and give you black and white vision using a pigment called rhodopsin. Cone cells work in bright light and give you colour vision using photopsin. 3 types of cones exist for blue, green and red. Your brain blends their signals to create the full colour spectrum you perceive. Light follows a specific path through the eye, and you should know it in order. It passes through the conjunctiva, cornea, aqueous humor, lens, vitreous humor, hits the retina, and focuses on the fovea centralis. The lens changes shape to focus at different distances. Aqueous humor maintains eye pressure and feeds the avascular cornea. Vitreous humor fills the back cavity and is never replaced. The eye has 2 fluid-filled chambers in front of the lens. The anterior chamber sits between the cornea and the iris, filled with aqueous humor. The posterior chamber sits between the iris and the lens, also filled with aqueous humor. Aqueous humor drains through the scleral venous sinus at the junction of the cornea and sclera. If drainage gets blocked, pressure builds, which is the basic mechanism behind glaucoma. 6 extrinsic muscles move each eye. The oculomotor nerve CN3 controls 4 of them: superior rectus, inferior rectus, medial rectus, and inferior oblique. The trochlear nerve CN4 controls the superior oblique. The abducens nerve CN6 controls the lateral rectus. If a patient cannot look sideways, you are thinking CN6 and the lateral rectus. Visual signals leave each eye through the optic nerve, which is CN2. At the optic chiasm, fibres from the nasal half of each retina cross to the opposite side. This partial crossing is what allows binocular vision, where your brain combines images from both eyes into a single 3-dimensional picture. The exam may ask where visual fibres cross, and the answer is always the optic chiasm. The lacrimal apparatus keeps your eyes moist. The lacrimal gland makes tears which flow across the eye and drain through the lacrimal punctum on each eyelid, into the lacrimal canals, through the lacrimal sac, and finally down the nasolacrimal duct into the nasal cavity. That is why your nose runs when you cry. On your exam, eye anatomy typically shows up in scenario questions. Someone loses peripheral vision, or cannot see colour, or has trouble focusing. Trace each symptom back to the structure. Colour problems point to cone cells. Night vision problems point to rods. Focus issues point to the lens or ciliary body. That reasoning chain is what they test. Now for hearing, the external ear collects sound with the auricle and funnels it through the external auditory meatus to the tympanic membrane. The middle ear has 3 ossicles, the malleus attached to the eardrum, the incus in the middle, and the stapes pressing against the oval window. These are the smallest bones in the body. The inner ear is where things get sophisticated. The cochlea handles hearing. Inside it, the organ of Corti sits on the basilar membrane and contains hair cells that are the actual auditory receptors. Sound vibrations move fluid in the cochlea, which bends these hair cells, generating nerve impulses sent through CN8 to the brain. Balance comes from 2 separate mechanisms. Static equilibrium detects your head position relative to gravity using structures called maculae in the vestibule. Dynamic equilibrium detects rotational movement using the crista ampullaris inside the ampullae of 3 semicircular canals. When your head rotates, endolymph drags over the crista, bending the cupula and stereocilia to send signals to your brain. The three semicircular ducts are oriented in different planes to detect rotation in every direction. The anterior duct is nearly vertical, the lateral duct is nearly horizontal, the posterior duct is vertical, but oriented differently from the anterior. Together with the maculae, these structures act like an internal gyroscope, telling your brain exactly where your body is in space. Remember that mastering these structures takes hands-on practice with real questions. The free practice questions are designed specifically to test whether you can trace a sound wave from the auricle to the brain, or explain which structure fails when someone loses their sense of balance. They match the OA format closely, and the link is in the description. Taste works through chemoreceptors on your tongue and other surfaces in the mouth and throat. 5 basic tastes exist: sweet, salty, sour, bitter, and umami, which is that savoury, meaty flavour. Tastant molecules dissolve in saliva and contact gustatory receptor cells inside taste buds. Each taste bud opens through a tiny taste pore in the tongue epithelium. The tongue has 4 types of bumps called lingual papillae. Vallate papillae are the largest, sitting in a V shape at the back. Foliate papillae line the sides toward the back. Fungiform papillae are scattered across the surface like small knobs. Filiform papillae cover most of the tongue, but here is the catch: they contain no taste buds at all. That last point is a favourite trick question. Inside each taste bud, 3 cell types work together. Gustatory receptor cells are the actual sensory cells that detect tastants. Supporting cells are long, spindle-shaped cells providing structural scaffolding. Basal cells sit at the bottom, acting as stem cells, constantly regenerating the other 2 types. Gustatory hairs extend from the receptor cells through the taste pore, increasing surface area exposed to whatever you just ate. Taste signals travel through 3 cranial nerves. CN7, the facial nerve, carries taste from the epiglottis and throat. CN9, the glossopharyngeal, carries taste from the vallate and foliate papillae. CN10, the vagus, carries taste from the fungiform papillae. The signal goes to the gustatory nucleus in the brainstem, then to the thalamus, then to the gustatory cortex in the parietal lobe. Smell uses the olfactory epithelium high up on the roof of the nasal cavity. Odorant molecules hit olfactory receptor cells whose cilia perform transduction, converting chemical signals into electrical impulses. These receptor cell axons pass through the cribriform plate, a bony sieve between the nasal cavity and the brain, and synapse in the olfactory bulbs at structures called glomeruli. From the olfactory bulbs, information travels through the olfactory tracts to the temporal lobes. This is CN1, the olfactory nerve. One clinically relevant detail is olfactory adaptation, where your nose stops noticing a sustained odour over time. The exam might describe a scenario where someone can no longer smell something and ask whether the receptor or the pathway adapted. As we wrap up, I want you to see how these 3 systems connect. The hypothalamus sits at the crossroads. It receives sensory input from the nervous system, controls the pituitary gland to regulate hormones, and processes signals from taste and smell through its connections to the limbic system. When you smell food and your mouth waters, that is all 3 systems working together in real time. For your exam, know the cranial nerves that serve each sense. CN1 for smell, CN2 for vision, CN7, 9 and 10 for taste, CN8 for hearing and balance. Know the 3 meninges in order. Know the difference between water-soluble and lipid-soluble hormone signalling. Know the 5 anterior pituitary cell types and their hormones. These are your high-yield targets. The free practice questions in the description cover every single concept we discussed today. They are completely free, and they are structured to match the OA exam format. Working through them after watching this video is the best way to turn what you just heard into points on your exam. You have covered a lot of ground today: the nervous system, the endocrine system, vision, hearing, balance, taste and smell. These systems are not isolated chapters in a textbook. They are a single communication network running your entire body. Understand the connections between them, and the exam questions will feel like conversations, not riddles. You have got this.